Hi, everyone. Thank you so much for your time and thank you for having me. And thanks, Katie, for the introduction. And I would just like to add to that my journey with corn production and corn agronomy began when I was doing my grad school training at Michigan State. So I just moved across the lake from Michigan, and I would say I'm having a wonderful time experiencing the similar winter. So that doesn't make me miss Michigan much. So today we'll be talking a little bit about the plant pathogen interactions or the systems approach that we can use to incorporate in our corn production systems. Corn is not generally a crop that comes to a lot of people's mind when we talk about organic systems or sustainability in that respect. But there are a lot of moving pieces to corn production systems that can actually be harnessed to look at the production systems from a holistic approach, as Katie mentioned, and make profitable management strategies and economical decisions. So before we go a little bit more into the depth, I would just like to highlight some of the corn production stats from 2024. So these are the state estimates from 2024, and we started off the year with one of the highest planted acres this year, and this number encompasses both the conventional corn and the organic corn. However, we did have a very interesting season this year where we started off really wet, and then we had a dry spell during the fall, and that's why our harvested acres for grain drastically fell. Our production numbers were also lower than what we saw in 2023, with the overall production being 7% less than last year, and even our yield numbers were lower than last year, about 2 bushels per acre on average. For silage corn, which is the whole plant corn, where we harvest the whole plant at about 65% moisture, over there we also saw a decline in our numbers. So when we look at corn production in the state of Wisconsin, it is a very huge economic program going on. However, our organic systems do not make as bigger part of it, but they are considerably still very large if we compare it with the overall United States. Our state stands at about 6th position when it comes to the production of organic corn, and for silage, it comes at the top 1 position. So currently, as of the NAS 2022 estimates, these reports haven't been updated, unfortunately, as of yet. We had about 600 certified farms for grain corn, and we have about 250 for silage, which translates roughly into 29,000 acres for grain, providing us 3.4 million bushels of yield and then about 20 million in our GDP. For silage, we have about 1/3 of the total acres from grain, and it's around 10,000 acres, which give us the overall yield of 174K tons and give us the revenue of about $500,000. So now when we look at organic production, we look at conventional production, everything comes together in a system. And these systems work in interactions. So if we look at these interactions, these are actually impacted by the variables that are present when we have to make decisions for our corn production, right from the pre-plant, where we have variables like hybrid selection, and when we look at organic corn, it becomes even more critical if we need to, or if we want to include certain traits, or if we want to go for hybrids or open pollinated varieties. We also have planting date decisions to make, because it makes our plant exposed to certain kind of conditions during the season. For our plant populations, that's also very critical decision when it comes to specifically organic corn, because we do have limitations for chemical fertilizers that we can enter to our system. During the growing season, we have other We have other decisions, other variables going on where we have fertilizer management, we have pest management, we have irrigation, rain-fed systems. We'll be talking about some of these in a bit more detail as we move forward. And we end up with harvest, which is also affected a lot by what are we seeing around and how it is impacting the time of harvest, how is it impacting our dry down moistures and our storage conditions. All these variables actually impact how our plant will respond or be susceptible to the challenges that we see in the Great Lakes region or in the upper Midwest. So starting off, we have a shorter growing season. We have limited number of frost-free days, and that limits the relative maturities of the hybrids that we use to a greater extent and also impact our overall yield numbers. This also comes along with variable climate, and over here the map that I have shows the percentage of precipitation as it deviated from normal during this year. To look at it, it looks like a very normal to a little bit of a wetter season, but what was the challenging part was the variability or the intensity with which we got rainfalls at the beginning of the season and then the dry spells that we got during the silking stage of the core. All these conditions, early season drought or humid and wet conditions, etc., result in further challenges which bring up certain plant pathogen problems, the major ones being ear-end stalk infections, foliar infections, and I assume a lot of corn growers, if we have any in this room, would have seen southern rust pop up in their fields this year. And that was actually a very interesting interaction if we think about it, because we had a mild winter last year and some of the southern rust pest kind of moved up north, overwintered, and it showed its impact during our season. These fungal infections or the pathogen interactions are always aided by the favorable environment and secondly by other external factors such as ear-damaging insects. All these challenges in collaboration, or I would say when they come together, they result in not only in yield losses, but also in quality concerns, and that amount to quality concerns in terms of test weights, in terms of digestibility, in terms of the toxin accumulation in our produce. So when we look at the variables of corn production, when we look at the challenges of corn production, and when we look at the environment that surrounds it, that's where the interactions come into play. So for anything to occur, all these things have to come into play in a way that we have a susceptible host or we have a plant which is then present in an environment which may aid the proliferation of a certain kind of pathogen, which can additionally be aided by the presence of other external factors. Those external factors can be anything ranging from insect damage to a hail damage, which is, leading your plant lose its turgidity, or also eventually, I would say animal damage. Deer damage is very common sometimes in the countryside for upper Midwest. So when we look at this and we look at the Great Lakes region, the major interactions or the major challenges that we see stem from ear rots, pink ear mold, white ear mold, green ear molds. We also see certain stalk rots and stalk boring insects, and these are becoming actually an additional challenge these days with certain reported instances of resistance against the present insecticide traits in our corn hybrids. And then we also see interactions for viruses which are transmitted through various insects. For example, I have a picture of a corn leaf with aphids on it, and I have learned that few years ago it was a great deal of an issue where we had higher flights for aphids, and we had issues with mosaic viruses. Today or currently, what we are seeing mostly are the top three, where we have Fusarium earrot, we have Penicillium, we have additional threat or coming up threat for stalk rots and stalk boring insects. All these infections not only impact the overall plant health in terms of yield, but they also produce certain kind of compounds that are known as mycotoxins, translating into toxins produced by fungi. These compounds, they render the plant or render the produce unfit for consumption. And the two most recent incidents of mycotoxins in the Great Lakes region have been observed in 2018, when we had about more than 60% tested samples across the upper Midwest, including certain parts of Iowa, having higher than 1 ppm vomitoxins. We also, so one of my colleagues back in Michigan State, she did a survey across the state, and she found that a very high number of samples had more than 10 mycotoxins present in a single sample. And we saw something similar in 2023, where our tested grain samples and our tested silage samples had very high accumulation of toxins in that. And the thumb of Michigan, which I would explain a little bit into further slides, had a higher concentration as compared to the entire state. So since I've been talking about mycotoxins, a little bit about the background on why are we talking mycotoxins today and in what are we talking mycotoxins today. So as I mentioned earlier, mycotoxins are secondary metabolite compounds which are produced by ear mold causing fungi. When we have elevated mycotoxin concentrations, it results in feed refusals, especially in case of silage corn. So the picture you're seeing here is a silage field being harvested and silage being chopped down. And another challenge with silage corn is that a lot of silage corn in upper Midwest is fed on farm and is not actually tested for toxins as opposed to the grain corn. So in order to address all these knowledge gaps or loopholes and what we saw in 2018, by the time we were in 2023, we had certain answers and we had certain data. And how did we do that was we conducted certain silage corn grower surveys across the state of Michigan, where we collected samples from around the state, and that was accompanied by a survey sheet where we asked the growers to provide some of their field history and management decisions to us. The exchange was they would provide us a silage sample, they would provide us the information, we provide them an interpreted report, tested report and interpreted results for mycotoxin conditions in their field. And that's when I learned that if you give the growers back an actually useful information, the number of survey responses jump from say 33 to 75 in the next year. So that was a great learning actually. The results from our survey were very interesting, and we found that all the samples that we tested across the state had mycotoxins in them. There was not even a single sample where we didn't see at least one mycotoxin. And as I was talking about the thumb region of Michigan in my earlier slide, here also we saw that the thumb region and also the southwestern region of Michigan had a higher concentration of mycotoxins as opposed to the rest of the state. Now, why is that happening? That also takes us back to our systems approach. These regions that I'm pointing out or I'm talking about, they're surrounded by the lake. So the thumb region, or the Huron County as the topmost county over there, had the highest concentration, and it is surrounded by Lake Huron. The environmental conditions during the silking period of corn in that area have high relative humidity, and also the temperature conditions are around somewhere between 22 to 25 degrees Celsius, which translates into 72 to 75 Fahrenheit. I'm sorry, I'm originally from India, so I still think in Celsius, and that kind of, throws me off a little bit. So those environmental conditions actually give very nice cocktail for our fungi to grow, to prosperous, and impact our plants. Another interesting observation, as we were looking at my colleague's work in grain corn, and we were seeing multiple toxins, we saw that even in our samples, silage samples, we had multiple mycotoxins present, and then we ran our correlations, and we found that the toxins that came from the same species of the fungi and also the ones which were isomers of each other were present together. So this means, so technically this doesn't really help a lot in management, but it can still help in identification of the problem and also lowering the overall cost of mycotoxin testing. So as we move forward, I would just like to remind everybody of this beautiful survey map or survey sheet that we circulated across the state. And what we got from this sheet were these numbers. So we found that our top three agronomic factors that impacted, or the top three factors in our entire cropping system that impacted our mycotoxin concentrations were crop rotation, planting date, and tillage. Although tillage or crop residue management wasn't significant, it was still showing us trends when the crop residue management was done in a way that we removed most of the infected residue from the field. The concentration and number of toxins found were lower. If we look closely to the crop rotation, it was found that the corn that followed a host crop of fusarium especially had a higher concentration and number of toxins as opposed to the ones where we had a non-host crop and a corn rotation. Similarly in planting date. So if we look at this chart, the mid-planting window is the window when most of the growers in Michigan, and very similarly in Wisconsin, I would say for Wisconsin it's a little bit earlier, the planting starts a little bit earlier as opposed to Michigan. They plant silage in that window. But that window is actually resonating a lot with the higher concentration of mycotoxins. And why is that happening? We'll go into a little bit more detail later on in this, because we also confirmed these observations with small plot trials whose data I'll be sharing soon. So when we were looking at these numbers, or when we were looking at this data from across the state, there were still a lot of unanswered questions. Remember, I was just talking about pre-plant decisions with hybrid selection being one, but there's no hybrid selection here because a lot of growers were using one kind of hybrids or one kind of traits. So the responses were so skewed that statistically it didn't make sense to run those numbers. Therefore, we decided to do certain systems-based management trials where we had a combination of hybrid insect protection and fungicide application. And I know this crowd does not believe in fungicide application, but just bear with me a little bit. So our trials had differential levels of insect protection in our hybrids, where we had hybrids with no insect protection traits. We had hybrids where we only were providing insect protection against European corn borer, and we had the ones where we were targeting both European corn borer and western bean cutworm. Western bean cutworm is another pest which became very prominent after we were controlling the European corn borer, because with increased use of Bt traits to control European corn borer, the broad spectrum insecticide consumption or spraying or application went down, and then a secondary pest was like, hey, I'm here. So now it's one of the major concerns for the corn growers in the upper Midwest and Ontario region. We also had our fungicide treatment with 2 levels of prothioconazole, and the results from these trials showed us very interesting correlations and made our belief stronger that if we approach the corn management from a systems point of view, we can actually reduce the reliance on chemicals, and we can improve our production and quality of the produce. So we saw that when we looked at insect feeding incidence, which was the percentage of ears damaged in each plot, and we also looked at the insect feeding severity, which is the number of kernels damaged in each ear, or percentage of kernels damaged in each ear. We saw that when we had the 2 insect protection traits, it helped control insect damage, which actually translated into the fact that we weren't seeing much of European corn borers. So the hybrids which were resistant against those, they were not performing any different. They were still susceptible to Western bean cutworm. But the hybrids with Western bean cutworm resistance had lower insect feeding incidence and insect feeding severity as well. This also translated into us seeing lower ear rot severity, where again, with the incorporation of insect protection trait, we were seeing lower severity of ear rots. And how is this possible? Because when we were preventing insect injury to the ears, the channels of fungal infection were reducing. So we were adding an insect protection layer, but it was actually protecting our plant from overall damage in terms of ear rot severity. We also explored these correlations, and we found there were strong correlations observed between ear rot incidence and insect feeding incidence, and this was further validating the responses that we were seeing separately in the earlier 2 slides. When we looked at the mycotoxins, the trend was very similar. We saw that the use of insect protection trait reduced mycotoxin accumulation across the site ears. So the site ear that I highlighted specifically is because that was the only one where we had significant amount of zearalenone as well. But we still saw similar responses across the state. So the correlations for insect feeding and ear rot with mycotoxins also showed us significant correlations. These, unfortunately, the R-square numbers were low, and it was a little bit disappointing. But the reason behind that was that we were overall seeing very lower concentration of mycotoxins in our small plot trials in our research stations. So in a year where we see a high concentration of mycotoxins, these numbers would actually make more sense. So as we are talking about the overall system, we were talking about the plant. We incorporated resistance in plant, but we are also looking at what the plant or which environment the plant is in. And how do we look at that? We look at how the environment around the plant is at a certain crop growth stage, and how the overall conditions are in the field during a certain crop stage. In order to study that, we conducted another set of trials where we had planting date and seeding rate as our treatments, and we had multi-location trials again for this. And these trials also consolidated our results that we observed from our growers survey, where we saw that the silage that was planted in the mid-planting window had higher insect feeding injury and ear rot, and higher ear rot injury as well. The correlations were very clear when we observed it overlaying each other. And we also saw that mycotoxins, DON was the most frequently occurring mycotoxin, and the highest number of samples which had DON greater than 1 ppm or 1 microgram were coming from the mid-planted silage. We also think that repeating these trials in another year where the concentrations are more higher, it would provide us a little bit more insight into these systems. But if we look at that, and I think I kind of talked about that a little bit earlier as well, what was happening in the mid-planted silage? So for the mid-planted silage, when we planted between 11th May to 30th May for Michigan or for Upper Midwest, it generally typically silks around the end of July to mid of August. And that's the time when we have the higher number or higher levels of precipitation levels in a typical year. Remember, 2024, very different, very dry. September, we didn't see much. There were a lot of other concerns. But if you look at 2023, and if you try to remember, later June, July, we were seeing a lot of rainfall. And that was a little bit more closer to a typical rainfall pattern for that part of the year. 2023 also started with the drought, so it had its own challenges with that too. So what I'm trying to explain here is that when we plant silage between the mid-planted window, the silking falls into the time when the environment around the plant is very conducive to the infections by Fusarium. And if we have insect flights coinciding with that, which is typically what happens with Western bean cutworm life cycle, the injury and the availability of fungus coincide, which creates a nice cocktail of disease and mycotoxins going forward. We also looked at the yield numbers, and as expected, the mid-planting had the lower numbers because we were seeing the damage. We also looked at the overall yield and nutritive value for silage because this was a silage crop. So we wanted to look at the fiber content. We wanted to look at the digestibility. We also wanted to look at the predicted milk yields. I'm sorry, there are a lot of numbers here, but I would just like you to focus on these two boxes where we saw that although early and late planted silage had lesser disease, lesser insect damage, lesser mycotoxins, the early planted silage had a better quality in terms of nutritive value for the growers. And why would that be? Because it gets an appropriate amount of time to actually reach the dry down levels, and the harvesting time is not a constraint during those scenarios. Further moving along, we were also interested in seeing how the in-field environment or the plant-to-plant environment affects the overall insect and ear rot severity in the field. And we found that as we were increasing the plant population, we were seeing a higher insect severity, higher insect feeding severity, and higher ear rot severity. So why would this be happening? We were sitting down and we were like trying to, contemplate why are we seeing a higher insect damage or higher ear rot damage in higher populations. So when we have a higher population density in the field, there is two things happening. One, the plant-to-plant competition is higher. Therefore, the overall health of the plant is not as prolific as in a lower density it would be. Secondly, the overall moisture levels or humidity levels are higher in those conditions, and it kind of gives that nice environment that the fungus wants. And third, the key pest that we were seeing in terms of Western bean cutworm has a habit of mobility. So when the plants are closer to each other, the mobility of the larvae is easier. So one larvae was actually damaging more plants, or it was causing more damage as opposed to what it would do in a field where we have a lower plant density. We also saw that our forage yield numbers were not increasing, I would say, linearly. There was more of a quadratic relation. And the most interesting stuff that we saw was in our nutritive value, where with an increased seeding rate, we saw an increase in the fiber content of the silage component for the silage component, which was actually rendering the nutritive value lower as compared to the lower or the lesser denser populations. We also saw that the starch content had increased up to a certain level, and then we saw a sharp decline. The other interesting observation was from the digestibility, where we saw a linear decline in the digestibility as we increased the seeding rate for our populations. So overall, all these experiments, when we look at them together, it comes across that when we manage mycotoxins or when we manage ear rots, there is no one single silver bullet. It all has to come together as a systems approach, where we try to protect our plant from the exposure it's having at a certain susceptible stage from the environment. We incorporate certain inherent resistance to it. We incorporate some additional control measures, where we are controlling or we are deciding on incorporating certain kinds of fungicides or biologicals, where the overall protective environment is created. So with that, the overall takeaways from the research that was done were that insect feeding was directly increasing insect feeding was directly increasing fungal infection. Yield and nutritive value were impacted when we saw a higher amount of insect injury, higher fungal infections as well. And in order to manage those, the most beneficial options that came up were incorporating hybrid insect protections. It was also changing or managing planting times, managing plant populations as well. And the last thing that I would like to point out is that you might have seen that across all the slides, there was one thing that was very consistent, which I didn't actually speak of, was field scouting. So when we scout fields, it's always easier to identify the problem first and then apply our timely management strategies, or also learn from previous year's scouting and then apply those learnings for the management decisions that we are going to take for the next year. So with that, I would just like to highlight what some of our focuses are going forward with the Corn Agronomy Lab. So we have this ambitious aim of actually evaluating various insect pests across the state with conventional and organic systems, using and leveraging our corn performance trials. We're hoping to get certain collaborations with plant pathology and entomology to realize those ambitions. We're also looking to work with Dr. Emily Bick in future with understanding Bt trait resistance to European corn borer. And then with the short corn becoming a more emerging product or becoming a more novel concept in corn production, we are trying to look at the susceptibility and resistant traits in that, and also how would its feeding preferences will be for dairy and feed cattle. And before I end with the future focuses, I would like to highlight what we have already been doing in the Corn Agronomy Lab and what has been happening with my predecessor, although he retired last year. But there have been a history with Corn Agronomy Lab where we do corn evaluation trials, and we make sure we have certain or at least some of corn or organic corn hybrids which we test across the state. So these are the numbers from 2024 for South Central region. The highest performing hybrids are highlighted, and we provide all the information beginning from yield to performance index to any insect pests and we aim to provide any information on insect pests being observed. Similar observations we are conducting in the other part of the states. Here we have the north central region of Wisconsin, and these trials actually form the part of a bigger effort, which is the corn hybrid performance evaluation trials. And the numbers on the Wisconsin map actually show the average yield numbers that we got in our corn evaluation trials. These numbers look a lot higher than the state estimates, and that's because we are dealing with corn at much smaller scale, because we are more focused on hybrid testing as opposed to overall production under this effort. So if you scan this QR code, it will take you to the booklet, where you can explore this information more explicitly. And I kind of circulated some of the forms in the crowd. So those are the forms that we use to actually get the application entries for these trials. Unfortunately, for last few years, our organic corn trial entries have been going down. Not much market interest have been shown in order to get those corn hybrids tested. So if you have a organic corn hybrid which you would like to get tested in across the state of Wisconsin, or a friend or a friend of friend who has a corn hybrid they want to get tested, please reach out to us, Corn Agronomy, and we'll make sure we include your trials in our overall effort. And with that, another thing that I would like to advertise a little bit is the corn management survey for Wisconsin corn growers. So this targets both the conventional and the organic corn growers. Once again, I'm trying to apply my learnings from Michigan in Wisconsin, where we're trying to collect information and areas of research interest from the corn growers and actually identify knowledge gaps to bring the useful research to the growers right to their doorsteps, sorry. And with that, I think I can maybe have it here for another 5 seconds if anybody wants to scan this. Oops, sorry. Yeah. And with that, I would like to thank all my collaborators, my project investigators, and other wonderful technicians that helped me do all this work, and I would take any questions that you may have. Thank you.
Can you explain how this information can be applied to Indian corn, native corn?
Yeah, okay. So for that, I think the key process is to actually avoid the stresses around the plant. And I think the key component there would be managing planting dates in a way that the susceptible growth stages do not fall when we have the most favorable environmental conditions for insect flights and fungal infections. And secondly, not shooting for very high populations and also managing them in a way that it doesn't compromise our corn yields, but also maintain the overall plant health and the environment, and not make the environment very conducive for infections. I hope that answers the question. Thanks.
I would also love to hear from you all. I think we have 15 more minutes. If there's any research questions that you would like to see us address around organic corn production, what are some of the issues you're facing that you'd love more research for or answers? And also, if there's just generally questions about organic corn production, there's a lot of knowledge in this room with other farmers. So if you have other questions that you would like to kind of crowdsource some answers to, you can kind of open up more broadly for organic corn production.
Yes, please.
A question about corn population, planted population. On our farm, we typically plant a flat rate population, making the assumption that in the organic system that we're going to reduce that population through mechanical weed control. When you were establishing your planting populations, were you going off of the recommendation from the seed supplier, or how were you determining the appropriate or ideal population?
So since these trials were conducted in Michigan, so I was going mostly by the recommendations that were available through the Michigan State University. And since I was looking at the silage plant populations, those are typically higher than the grain populations. So that was my go-to source to get those numbers from.
As a follow-up, would you say that in the grain system, that population perhaps is less important for mitigating disease pressures?
So I wouldn't say that, but because in grain system we have another challenge because it stays in the ground for longer, so it gives more time for mycotoxin accumulation. Because once your plant is infected with fungus and mycotoxins, the more time it has to, it has to stay in the ground, the accumulation kind of increases. So that's actually one other management technique that a lot of grain growers kind of, we kind of advise them to do so that they harvest earlier, so it's not accumulated until later in the season. But it's still important, and it's unfortunately it's not comparable. I don't really think I can give you a straightforward answer for that. So.
Have you been seeing differences with any of the trials that you've been doing with plant population? Do that balance between, like you said, plant population and increasing it in organic to take out the risk of cultivator blight, to have that higher density for weed management, then creating a microclimate that might be more conducive to fungal diseases. Have you seen anything on the farm? With those dynamics?
We're still learning.
Yeah. That sounds like, again, a research project kind of trying to, I don't know, particularly in organic systems with the genetics you have available, doing a study that is more systems-based, that looks at those interactions between weed suppression and risk of disease levels that we're seeing. Has anyone seen, go ahead, Eli.
Well, I'm just curious, and it was maybe up there, but what were the planting populations per acre? I saw by hectares.
Yeah, so we started off with 28,000, 34, 40, and 46. So we had like really low to high.
The increased insect feeding was in the 40 and 46?
Yes. Yes, please.
In the areas when you had map up in Michigan, I noticed where the mycotoxins or the toxins were the highest were close to water.
Yes, they were.
I'm wondering if you varied maturity at all. Usually, if you're growing a corn with maximum maturity for the region, it's going to pollinate at the worst time of year, the hottest, most humid, and you're close to water.
Right.
If you went with an earlier variety of corn and pollinated perhaps before peak heat and humidity, would that make a difference on disease?
I would think so, and that's a very good suggestion. And I wish, if I have resources in Wisconsin, I think I might want to, do something like that to actually look deeper into that. So for sure, I think planting early or using a maturity which kind of silks sooner or later than the actual or the most common time might actually help.
We did with Purdue University, and if you're interested in more of the details of the results, Kelly Devin, who you'll see at the registration table, she was the grad student that did the trials. But with Damon Smith from Plant-Pathology and Darcy Tulenko, looked at different biological fungicides, particularly for tar spot, as well as different genetics. And this was true when we looked at some of the biologicals also for Fusarium head blight and white mold. Is genetics is really one of our primary tools. I think the management with respect to populations and looking at altering that microenvironment is also important, but I think to continue some of the evaluation with respect to performance of organic cultivars, also in the context of organic management systems. So I think that's where there's sometimes some sticking points with some of the university trials, is that they're using organically available seed, but not putting it in the organic context.
Right.
One of the things that Chris Wilson stressed last night, and when he was talking about his very diverse rotation, is that can really change what you're seeing in terms of disease pressures. Very diverse rotations we use in organic. But do you do any evaluation yet at Albert Lee on some of the disease
On like
resistance?
under organic management
Yeah.
specifically? Unfortunately, no.
Yeah, that's a real.
Huge opportunity, but the same reason we use technically with conventional management.
Yeah.
That being said, there's a lot, everybody is trialing corn every year, right?
And there's a way to get some sort of a network of farmers.
To a certain degree, I figure you can draw correlations between that hormone.
Yeah, it's been a huge bottleneck to do really good variety trials in organic systems. Because again, that changes the dynamics of what we're seeing. Maybe there's another thing to put on the dream list is to create some sort of on-farm network of variety trials that we can kind of crowdsource some of this data. To speak of that, so on the vegetable side of things, there is, and I don't know if anyone here is involved in this, it's a platform called Seedling, and this is exactly what that's for, is to, and it's for organic vegetable production, but it crowdsources information from on-farm trials that really focuses on the traits that are important to the growers. So not necessarily diving into kind of the depth of data that we typically take at the university, but really focusing on what farmers want and need to know in terms of variety selection. So we should look into whether that would be a platform that this group might be interested in trying to crowdsource some of this information in terms of performance and disease resistance under different plant populations, different rotations, different latitudes. So yeah, I think Julia, if the issue of trying to get good data from organic systems is definitely a struggle.
Yes, please.
Has there been much effort to see about actually purposely damaging the plant mechanically and see what kind of...
I'm so glad you asked that because that's another trial we have been doing. I didn't include it in here. There are actually two types of damages that so some people have already done that in Ontario, where they were actually going in the field and they were damaging the corn ear. But what we have been doing in Michigan, and now actually we did that in Wisconsin this year as well for our Lincoln location. We were going in the field and purposely chopping off the leaves at certain stages to simulate leaf damage. And we were aware that it is not as consistent with what we would be seeing in case of a spotty disease or something. So we also tried using certain defoliation chemicals for that. So we were spraying those defoliation chemicals, which would actually burn the leaf on contact. So kind of simulating the leaf damage on the spot affected. Unfortunately, I have not analyzed that data as of yet. It's a mine of data that's kind of sitting in. So hopefully sometime down this year, we will have that information out. So, yes, please.
Yeah, it's really see your research. One thing that I'm a corn breeder and I've been breeding corn for 36 years under organic conditions. And when we get conventional inbreds from plant variety protection, we come to the mill, and we start growing them under organic conditions and keeping the seed, we start to see a reduction in fusarium.
Oh.
And some of these XPVP things are really loaded. And looking at the literature and looking at our results, because we start finding that through breeding under organic conditions, and collecting in particular the plants that respond, that don't need fertilizer,
Right.
Fertilizer, we see that the plants are accumulating endophytes in the tissues. And we think these endophytes are very important for controlling fusarium. And that's what we see. We see real decline in fusarium, particularly seedlings, because as a breeder, you're always looking to seed. And, you start out with some of these varieties that are absolutely horrible. But if you continue to develop them, or grow them, and select them, fusarium becomes less of an issue. And so if you think about it, people, almost all of us are using varieties that have been developed under conventional systems. Fusarium has this effect in the plant of reducing nitrogen efficiency in the plant.
Yeah.
Look like they're nitrogen deficient. And so what happens with the conventional breeders, they don't care. They add nitrogen fertilizer.
Yeah.
But when we start working organically to breed our crops, and we start selecting for the plants that do well without fertilizer, we're inadvertently selecting against Fusarium as a dominant endophyte. So Fusarium is a very strong endophyte. It's passed from seed to seed to seed to seed. And it's something that we really want to regulate. But I think that once again leads us to the question, we're starting to breed under organic
Right.
seed that we need. And for the plants that have that built-in ability to put Fusarium into place, not necessarily kill it, but make it less toxic to the plant.
Yeah.
I hope that
No, yeah. No, I'm really glad you brought that up, because when I was a student, I came across some literature on endophytes, and I was trying to convince my PI that we should write a project on this. But I mean, there were a lot of logistic issues. But yes, I completely agree with, and thank you so much for suggesting that.
We should talk about that, because as we're starting, we have a grad student, but we haven't got applied for a grant proposal yet. From just what you're saying, Walter, from what I found in the literature, and some of this, I was giving a biological seed treatment talk to the American Seed Trade Association a couple of years ago, and diving into some of the literature related to biological seed treatments. And some of the endophyte seed treatments, there's some really compelling literature to show that interaction with the endophytes can be, again, it's not going to be like a all or nothing treatment, but that can be quite powerful in terms of reducing the Fusarium load. I was looking at it more from the context of food-grade wheat production, but that's interesting too in the context of corn production. We haven't done work in corn, but we have done work in carrots as a model system to look at older open-pollinated varieties of carrot versus more modern inbreds. And this isn't just with carrot, but it's been shown over multiple crops that the phenomenon that Walter was talking about, we do indeed see that as we've bred under conventional conditions, these elite hybrids, they have moved into having less, I wouldn't say ability, but they're less apt to make these associations with soil biology because it historically has been bred out because there's been a cost to the plant, which is okay if we're dumping on synthetic nitrogen. We know now of all the issues that causes in terms of overall plant health and the plant's ability to withstand insect and disease pressure, but that is a phenomenon that's been well documented. So when we look at the role of these soil microbiome and endophytes and the loss of the propensity of the plant to have those interactions, we're losing a tool in organic systems to be able to create more of a holistic way to have plant health and those more natural mechanisms to resist diseases. So I'm super excited about, we're getting some isolates of these different endophytes from a lab in Virginia, and we're going to be testing those as essentially seed treatments to see if we can be able to create another tool to help the plant fight from the Fusarium as a pathogen.
That was actually one of the reasons we didn't go ahead with that, because we weren't able to ensure that we'll be having isolates, provided or anything. So.
Then you get into the issue of trying to translate the technology to stable seed treatment. I think some of the issue we've seen with the performance of biological seed treatments is the stability of the microorganism as a seed treatment. They're living organisms, so depending on the nature of the coating, how that has been stored, the time from when that seed treatment was produced, you're going to see variable interactions in the field. So to be able to look at how do we ensure consistent performance of these seed treatments that we're able to convey to the farmer what is going to be the level of potential success of using those treatments, it's been hard to show the stability and what to expect from year to year with those.
Under that We have to start thinking about moving away from inputs, which is another input. Maybe not
Yeah.
necessary one. I'm not saying that, but building plants that have these communities of microorganisms that help them under multiple stress conditions. And that takes a concerted effort and a different way of thinking about it.
Right.
So that we aren't necessarily thinking about a wonder product, a silver bullet, that you can sell it, somebody can sell it on and make money from, but rather having seeds that actually work, that where you need less input, and that the microbial community has maybe even coming from it, even coming from the region, is actually indigenous. And fostering its partnership with corn, particularly corn, and getting something that really belongs, really has roots. And I think that's where we should be focused.
There's a question at the back of the room. I'm sorry.
Another question about how you characterize the planting dates. Most organic farmers are planting their crops, let's say 2 weeks after their conventional neighbors, which puts us kind of in that mid category. From the data, would you characterize that as maybe a risky decision that you would maybe encourage to wane even further?
So delaying wouldn't be a very, I would say, favorable decision in a way that it will also compromise your harvest timings and those decisions as well. So that's a lifelong battle, I think. Even with if I'm in a room with soybean growers, then that's another challenge to say put corn first, soybean first. So I think maybe trying to avoid that window as much as possible could be the key role. Or as somebody else mentioned, like going for a relative maturity, which doesn't put us in the time frame when the silking kind of coincides with warm, humid, moist conditions, could be something to look forward to.
I believe Iowa did some research around tar spot, and it wasn't showing up on a lot of the organic acres, but it had a huge problem with conventional acres. And it was the application of manure. A really reduced tar spot. I noticed it where I live. And so I think it is microbiome.
Yeah.
Or trace elements, that make it
Yeah.
present in manure, but not in conventional fertilizer.
Yeah.
But if I'm not mistaken, there was some research done on that out of Iowa.
Interesting.
If it affected tar spot, I think it would probably affect more diseases as well.
Yeah.
Certainly, I think a healthy plant is going to be able to resist better pest pressure. So looking at balancing fertility, making sure you're addressing the fertility needs on your farm, making sure that there's not excess of nutrients that are going to cause issues as well, whether that's excess nitrogen, phosphorus, other issues. And that's again another area of research that we certainly need more work related to is understanding that aspect of the soil fertility system and that balanced nutrient, how much that confers better ability for this plant to withstand. So that's interesting with the manure component. Again, manure is definitely having positive impact on the soil biome.
Yes, please.
Assuming these tests were done on 30-inch rows, was there any experimentation with using a wider row spacing? How that would affect?
So unfortunately not, because we didn't have the planter equipment to do that. So if, going forward, we have a planter in the program which can actually, maneuver those settings, I think looking at that would be interesting.
And a 60-inch row system, you have better potentially airflow between the rows, but the way we've implemented that system is very high density within the row, and then stress the plant, and plant stress is then going to exacerbate disease and pest pressure, as well as potentially creating different microenvironments. That's a great question to look at.
Opens up weed pressure.
It does open up weed pressure.
Yeah.
Certainly, it's that's been a challenge with organic on those wider roads. But yes, you have wider roads, but they have increased density and stress within the road.
Have you seen any increase in the phytotoxins from a certain cover crop or even like in a corn on corn system? Do you expect phytotoxin residual to take up into that second year?
Yeah. So a lot of growers who were going corn on corn, and also even in fact who were following their wheat with corn, we did see those higher incidences, and that's primarily because those are the host crops and the inoculums like still in the field, and next year it kind of rejuvenates and then yeah.
Would using a latent or trying to incorporate more broadly cover crops maybe prevent that?
I would like to think so. I haven't looked into literature if there's an answer on that, and I haven't done any research myself to comment on that. But I think that could be a potential opportunity there. Yes.
So we're at time, everybody, but let's give a big round of applause to Dr. Aaron. Thank you so much.
Thank you so much.